All right, let's get started. Hello, and welcome to this webinar about discovery and development with LabQ Biologics LIMS. Thank you very much for being here today. My name is Bernie Lee. I'm a product manager here at LabQ Software. I have a short and sweet presentation for you today where we will talk for a few minutes, and then I'd like to show you some of the product. We'll be talking about some of the challenges of antibody discovery and development or similar, and the tools and strategies that we employ to drive success, improve efficiency, and arguably most importantly, increase the quality of life for scientists that are working hard in the lab. We'll be looking at some plate-based screening and characterization with analysis and reporting to illustrate a lot of those Biologics LIMS features. So the general contours of discovery and development commonly involve many of these kinds of steps, many stages across many specializations. In general, we screen libraries to identify potential candidates. We characterize and select the best through various assays. There's often an engineering step for examining liabilities and trying to improve developability. And ultimately, we develop reliable cell lines for producing our protein and then scale up its generation for various purposes. There is typically a great deal of iteration both within teams and across them throughout all of these obviously oversimplified processes. So for all of these various stages, they present many key challenges, key data and workflow challenges. I chose 3 to keep this simple. A high volume of data almost always results from these activities. They result in particular from things like iterative large-scale screens. So that's when you end up with many samples across many plates, across many assay runs, and trying to do all that kind of stuff into files makes manual management really kind of risky and time-consuming. Lots of errors in spreadsheets and the like. Then there's also integrating all of that data through that complex multi-step process that I mentioned before, from the various sequences or molecules or cell lines that you're working on to the various assays from multiple plates, that are performed, and then you have to take all of that and join it together in a way to perform the desired analyses that you want and to be able to make the decisions that you want to be able to make, particularly amongst groups. You make a lot of these decisions with a project team, and that can be really difficult to get it all stitched together properly in a complete, comprehensive, consistent, and reliable way. So then there's quality and organization. So this one presents all kinds of challenges, from limiting post hoc use and cross-team utility of your data, to introducing things like false positives, false negatives, things like that. Lots of challenges abound when you are working with such complex processes. And there are, of course, consequences to having these challenges present in your operations. So for starters, there's lost or confusing data. People lose track of samples. They lose data itself, or they can't make use of the data after the fact because of the way it's stored, because of inconsistencies between the way several people or groups have used these things, because a lot of the key pieces of information are in some PowerPoint somewhere that isn't predictably named, or it's because it's in a standalone ELN somewhere that was also poorly named and annotated, and you simply can't find that kind of stuff. Tracking back. So given that discovery and development often require multiple iterative screening rounds and handoffs between scientists and teams, it can be particularly difficult to track back from whatever stage you are, whatever step you're on, back upstream to wherever you have your primary plates that have the primary material on which you're truly doing your investigation. From those from those downstream plates and properly associate the information you have with the sequences or proteins that those samples represent. That's a truly painful spreadsheet game. And then, of course, there's loss of time, which is really probably the most precious asset that is being spent here. So these challenges cause scientists to have to redo experiments, dig for old data, reproduce material that's lost, very frequently reconstruct analyses and reports that we're sure that we've done before and submitted, but now we can't find it, so we have to recreate it from a set of files that we may also have to dig for. And what we really want to ensure is reproducibility and consistency across multiple assays and experimental setups as we go through all of this stuff. So what you want is a solution that provides at least the following, and almost certainly many more things. You can fill in the blank with what's in your head, but you want some kind of centralized application to manage your data, your samples, your analysis, including the ability to register and maintain uniqueness among the entities of interest that you are working with. You want some way of automatically integrating your entities with samples that you are actually tangibly working with, the plates that all of these things are on, assays, and the results that you ultimately generate for these things. You want to be able to generate analyses for hit selection and reporting, ideally without having to take data from here and combine it with data from there, and then copy those and stick into yet another application that you are then manually generating some plot or set of calculations for. You want an ability to document what you're doing, document your operations for that reproducibility and understanding in a way that isn't separated from your data. You don't want all of those different pieces of data and that analytical program and the need to take pieces from each of those things and put them in some standalone ELN. You're just adding layers to your problem. And of course, you want it to be easy, as I've sort of implied, to return to your data for insights in order to make additional decisions or to plan future work. And then, of course, you want something that helps you maintain the integrity of your data, kind of controls access to your data, and provides auditing of everything that you're doing so that when inevitable problems arise, you are able to go back and look at what happened, when, and where, and be able to reconstruct things that are important. Okay, like I said, I'm going to try to keep all this stuff short and sweet. We're going to jump over and I'm going to show you a little bit of this stuff. I'm going to show you some key aspects of LabKey's Biologics LIMS with an open invitation to set up a little bit more time with us to take a bit of a deeper dive. So our LabKey's Biologics LIMS is a powerful and flexible system that supports the discovery and development of things like antibodies. And today we're going to focus on a few key features. Generation of plates that we want to operate on, assay data capture accordingly, analysis performance, hit selection, a little bit of sample and plate management, and a little bit of bioregistry exploration to profile and track how known entities are generated throughout the process. So with that, let's jump over here. Yeah, there we go. Okay. So what you're seeing here is a set of samples. These are protein batches that are registered and stored in the system. This is an enriched data grid that shows information about the various batches that are present in the system. It includes information about how much of it you have, where these things are stored in freezers and things. And it also provides information about what these protein batches are related to. So in this case, this protein batch 5 comes from a molecule named M4, or this protein batch 4 comes from some Herceptin Fab material that we have produced. You can also see where some of that stuff came from. You can see what expression system or what constructs were used in the production of these things. All of this stuff ends up being nicely integrated. And another way of viewing that that I particularly like to use and show are these lineage graphs that we see. So here's my protein batch down here. And as I said before, we used this particular construct to produce this particular molecule. And you can really just see how these things come together. We ultimately got to this state by walking through, in this case, it looks like an initial immunization and some boosters for a particular mouse, extracting some spleen. So this looks like it was some kind of hybridoma workflow that had produced some cells from which we took the knowledge that we could, and then engineered a different cell line in particular to produce this molecule. And look, we got a batch of this. We got a batch of this protein. Similarly, I can look at some of these molecules in particular, and we can see that when we do come and we look at these, we can see how any data that I've gathered against these, in the same way that we can view them in this integrated fashion on our screen, we can also see any captured assay data against these particular molecules. So if I come up here and I click on assays, I can see that across all of the protein batches or purified antibodies of this Herceptin Fab, I have some assay data that contains some chromatography information, some page gel images, and some protein quantification. So coming back to our protein batches here, I just kind of want to jump into some of the actual action here. You can see that I have 5 of these protein batches selected that are available from our freezer. And what I want to do is, in this case, plate them. And I'm going to call this my demo characterization. And we are going to put these things onto some prefabricated plates, some pre-designed plates. So these are templates designed for this particular type of assay. Oops, what did I just do there? But what I want is I actually have several versions of this template that I want to make use of. And I'll show you what this looks like in just a second. But I'm taking these 5 samples and I'm distributing them across these plates. And because I designed my plates in advance, what ends up happening is that these samples end up getting distributed across these plates as dictated by my template. So ultimately here we have 4 plates that I produced that have these dilution curves of those particular samples of those particular protein batches distributed across the plates, and then similar but not exactly the same distribution across these other plates. This is our plate editor that allows you to both get a quality visual of what you're doing, as well as make updates accordingly. So I can see and click into the samples that are plated on here. I can examine these whole dilution curves, and I can make adjustments accordingly. Yes. So once I have these things plated and I want to actually run them through my instruments, we offer the ability to generate instrument instructions so that you can take these plates with the file, stick them on your instrument, get your plate reads back, and then import the data accordingly. So in this case, I want to show you this really fast. I have a multi-plate read that I spit out. This is big data, generic stuff, but it matches the 4 plates that I just generated. And it's just a luminescence read for what's coming in here. And whether your instruments produce a multi-plate read or 1 plate at a time, we can take these and ingest them automatically and produce the kinds of associations that we want. So coming back in here, I can import into this assay. It's as simple as dragging and dropping or selecting that particular file, hitting import and going. And what we'll find once this loads in just a moment is that the raw results from those plate reads in that plate shape, without me having to change anything on that file, was automatically associated here with the samples that were present that we were just looking at, and the wells for the different plates that are in the system. So right now I can see plate 3 here, but if I scroll down, you can see the rest of the various plates that I populated here, and their data accordingly. Here are the dilution curves that we were looking at before, and here are the results. So from here, I've also had a variety of important analytical components generated for me. So for example, I wasn't showing this by default, but we have, in this case, automatically generated a set of curve fits for all of the samples that were in this assay. I can see my EC50 values and mins and maxes and R-squared values and all sorts of other things for this particular chart, and make use of those in my analysis, my decision making, and my reporting. These charts are trivial to produce, to design, so that each time more runs of particular types of data come in, this sort of analysis is performed automatically for you. Okay. We're going to make a slight switch over to some of the more iterative operations that are supported here. We're going to go back into some of our plate sets here, and I'm going to demonstrate what it looks and feels like when you're doing something that's more iterative than just characterizing a particular batch of your protein. So what I have here is a primary plate set that contains a bunch of cells, and these are the materials that I'm interested in screening and characterizing in order to ultimately make some selections, and then progress them through the through the discovery process to where I can ultimately select some that I want to do some sequencing on, convert them over into stable cell lines, and perhaps get to the point that I was just showing you. Okay, so in the same way that I used some of those plate templates before to manage and populate materials that I put on these, I can do the same thing here. I've kept this relatively simple, where I take some of these materials in particular amounts and concentrations to start with, and then from here I can take various actions. So just to show you what some of this already looks like, and then I'm going to add to it, I have my primary plate, and I have pulled material out of there for a flow screen. But I would like to add to that. So in this case, I'm going to take this plate and I'm going to extract some material from it for running on ELISA. We'll give this a name. Let's see. Demo ELISA. Create this. And in this case, what we did is we pulled out a pre-configured quantity of the material at a particular concentration. And in this case, it's automatically populated with an antigen that I want to run this against. Now, in your template, you don't have to save, a particular antigen. You can choose what antigen you want to, you want to run your ELISAs against. In this case, I'm just kind of skipping ahead. And then just like before with the plate data that I imported, I can simply import my ELISA data and have it be populated. Now, one thing I want to point out really quickly is that for these iterative screens, characterizations, and selections, you can apply automatic hit selection criteria. In this case, I have a simple setup where I want to say, my positive binding, I want to be higher than 0.3, and the non-specific binding I want to be less than 0.1. Now, in this case, I've already actually imported some of this data. So what I want to do is simply show you that you can see from the additional line in this plate set hierarchy that I have pulled material from this original primary, upon which I can import some data, make some selections, and proceed forward. But I would actually like to show you that from the primary plate set. So you can see that I have my samples in their locations on my primary plate. And you can see some of these checkboxes over here. And these are hits selected from my ELISA results and separately on a separate plate from my flow results. So I can come in and I can look at those ELISA results. And you can see that by default here, I'm looking at data that is filtered for the hit selection criteria that I wanted. Now, I don't have to look at it that way. I can just start by looking at all of the raw data. And you'll note that we've automatically marked hits according to that selection criteria. And you can do things like view your basic bar charts on these things or heat maps. And of course, if you want to override those hit selections, you can override those hit selections. And once these things are marked based on the criteria that we want to make our selections, we can come back up and view those across multiple assays run. So again, I've taken some material from this original plate and I've run a flow screen before that, and then just a moment ago, I pulled some material out so that I could run an ELISA on these things. Regardless of the precise realism of this, the point is that the information from across these things can be swiftly combined. And I can say that I want only the hits that cover both of the types of characterization that I did. I want to select those, and I want to pick them from my primary plate. Again, they might be distributed differently on the full plate or the ELISA plate. I want to pick them from my primary plate. I want to move them forward onto, like a production ready material. Obviously, that's silly, but I'm going to just put these on a 96 well plate, and we're going to go. Okay, so here are the extracted samples, reformatted onto a new plate from which I can do whatever the next steps are. And you can see from this plate set hierarchy that this production ready material that has been through those various screens was pulled from this stuff, and we're ready to go. We can maintain those relationships and go back and explore them later after the fact. Okay. Trying to keep it simple today. The last thing or things that I want to point out are related to the sequences, molecules, cell lines, libraries, constructs that you produce in this system, and how we help you build and maintain some of those. So I showed you before that this Herceptin fab, I think is what we were looking at. As you work through the system doing the kinds of things that I was just showing you, we generate these relationships for you. And we roll data that you've generated at the bottom here back up to the top, and you can take a look at some of that quantitation. These molecules are unique in the system. They're real and regular in the system. They have identities based on their components. So here's a Herceptin fab light chain sequence that in this case, in advance, registered in the system, and it was recognized as such upon registration, based upon in this case, the kappa variable that's present, as well as this kappa constant immunoglobulin domain. that is here in the system. These automatically have a bunch of classifications and structural numbering things done. It has physical properties calculated for them that you can see here. And the same thing is true of the molecule that they produce, in that you can see those sequence-based physical property calculations and update those accordingly. So there are also a number of other things that I'm not going to spend a lot of time diving into today, but just to call them out briefly, there are we show you your inventory of samples that are related to any of these entities in the system, whether it's a particular protein sequence chain, a molecule, a set of cell banks, anywhere where you've written up some of these operations as you proceed through them. In this case, you can see there's a molecule characterization notebook that's built into the system. We're not going to spend a lot of time on ELN today, but all of those things are shown, and they allow kind of a bidirectional relationship so that you can both view and navigate how all of this stuff is put together. Okay, sorry about that. So fundamentally, the cost of discovering and developing a biologic is really driven by the amount of energy required by the people and the instruments that go into it. And I just showed you a few aspects of our Biologics LIMS, which reduce that energy required for a whole institution, as well as the individuals that are involved, driving efficiency gains and streamlining the discovery and development process. And so you end up with reduced dependency on spreadsheets, and a centralization that ultimately reduces errors and time required to track and report on your operations. You can track and maintain uniqueness for entities of interest through iterative plate operations while integrating all of that data for decision making. Again, you also end up with fewer lost samples, fewer repeated experiments. Much of the data capture and analysis can be automated. I showed you a little bit of that in terms of the simplicity of data import, but you can actually have LabKey listen for new data and automatically import it, as well as what I showed you, which was throwing your plate reads and other results at the system, and we will automatically make the proper associations, as well as generate critical calculations and visualizations that are needed for your analysis and selection. Again, I didn't spend any real time on it today, but there's also a data connected ELN that's built in so that in the same system in which you are performing all of your work, you can comprehensively document your findings. And what you end up with are fewer regenerated reports, happier people in bioinformatics and data science, happier scientists, happier managers, happier C-level and VP level personnel. And ultimately, what we find as we help people implement this is that the cost of using LabKey is lower than not using it because of how much time people spend on this other kind of stuff. So with that, I just want to thank you again very much for joining us in this hopefully simple and initial tour of LabKey's Biologics LIMS. I would like to dive deeper with you, learn more about your use cases, pain points, goals. So please keep an eye out for an email from me that has a link for scheduling a little bit of time. And I'd love to work with you to prepare a more tailored demo for your organization. With that, if anybody has any questions they'd like to ask publicly here, I'd be delighted to try to address a few of those. And if not, hopefully we can save some of those for a future discussion. Thank you very much. Oh, and maybe I didn't say it originally, but there's a Q&A feature in the Zoom that you can find at the bottom there if you want to add some questions. And I'll hang out here for a couple minutes in case any of those come in. I see. Okay, so the question here is somewhat related. There's a question about things like gene expression matrices and whether some of the capabilities that I showed will work for those. So, certainly to the extent that the way that you'd arrange your samples in plates and kind of iterate through the process, absolutely. As for the actual gene expression results, depending on how those look, yes, we can display some of those things and help you make use of them. In my experience, there are some of those gene expression matrices that require a little bit of reformatting or summarizing in order to make them, particularly readable to an end user. We can probably help with that. But I would, before I committed anything like that, I would want to make sure that I, heard more specifically about what you're trying to capture. But we do allow you to do a lot of data processing as you import your files against plates or other samples in the system. And we've helped a lot of people with some fairly sophisticated stuff. So I'd certainly like to hear more about that, and we can figure out how well we can support what you're referring to. Great, thank you. I'll hang out for probably one more minute, and otherwise I'd be delighted to, again, circle up with people and have a deeper conversation sometime in the near future.